First of all, I would like to thank the organizers to put this meeting together. So my lab studied how hypoxia regulates tumor progression and initiation. So today I'm going to share some of our recent studies. So we know, everybody knows that oxygen, and the oxygen is very important for our life and it's required for our life. And so the most important physiological function of oxygen is to generate ATP in the mitochondria. So which drives many important physiological processes. For example, the metabolism, exercise, and immune response, and respiration. And in addition to its physiological function, oxygen is also associated with many human diseases, including anemia, cancer, stroke, wound healing, and heart disease. So today my talk will mainly focus on cancer. So the solid tumor is hypoxic, which is due to imbalance of oxygen homeostasis. So the cancer cells grow rapidly and consume a large number of oxygen. On the other hand, the blood vessel within the tumor has a reduced ability in oxygen delivery and diffusion. So when oxygen demand exceeds oxygen supply, which causes hypoxia in the tumor. So the area next to the blood vessel is relatively oxygenated, and the area far from the blood vessel is hypoxic. And we know that some also the necrotic region at the tumor center, which where has no oxygen at all. So I just give you an example. In the breast tumor, the median oxygen level is about 10 millimeter mercury, which is significantly lower than that in the normal breast tissue, which is 65 millimeter mercury. So this is not unique to the breast tumor, and also, so as I said, it's hypoxia is observed in all the solid tumors. So as a result, the hypoxia-inducible factor, also known as HIF, is highly activated. And HIF is a master regulator to control the adaptive response to hypoxia. So HIF is a heterodimeric transcription factor consisting of alpha and beta subunits. So far, 3 alpha subunits and 2 beta subunits have been identified in mammals. HIF1 alpha and HIF2 alpha are the 2 most studied HIF family members in human cancers. So from the protein structure, you can appreciate that how similarity between HIF1 alpha and HIF2 alpha. So it's not surprising they share many common functions in response to hypoxia. So the function of HIF3 alpha is largely unknown, mainly because there's so many distinct the splicing isoforms have been shown in the cancers. So the HIF is HIF binds to the hypoxia response element to enhance over 1,000 downstream target genes in cancer cells. And those target genes are involved in many important cancer biology, for example, angiogenesis and metabolic reprogramming, epigenetic reprogramming, invasion metastasis, and also the cancer stem cell maintenance. So the HIF is considered a key driver of human cancer progression. So HIF activity is regulated by oxygen. So under normoxic conditions, HIF alpha subunit is hydroxylated on 2 protein residues by a family of protein hydroxylases called PHD. So this hydroxylation requires the oxygen as a substrate. So the hydroxylate HIF alpha is recognized by ubiquitin E3 ligase VHL. So this VHL can mediate HIF1 alpha degradation in the proteasome. Under hypoxia, the hydroxylase activity is blocked. So the HIF alpha subunit is stabilized and can dimerize with HIF1 beta to enhance the downstream target gene expression. So the HIF activity is also regulated by another dioxygenase called FIQ1, which catalyzes the asparagine hydroxylation. And this hydroxylation can block the histone acetyltransferases P300 CBP binding, so can inhibit the HIF-dependent gene transactivation. Again, under hypoxia, this asparagine hydroxylation is blocked, so the P300 CBP can bind to this transactivation domain to enhance the HIF transactivation. So in the past, this 30 years, and many studies have identified this extrinsic factors as well as intrinsic factors that control the HIF transcriptional activity. So besides oxygen, nutrients are also known to control the HIF transcriptional activity. And many intrinsic factors, as I just talked about, the hydroxylation and the ubiquitination play a key role in the HIF-1 alpha protein stability. And HIF transactivation also requires epigenetic regulators to modulate the chromatin accessibility. So the hydroxylation requires the alpha ketoglutarate as a cofactor. So many metabolites can affect the alpha ketoglutarate, can also affect the HIF protein stability and the transcriptional activity. Finally, the oncogenes and tumor suppressors, for example, mTOR or VHL or IDH, can control the HIF and transcriptional activity. So today I just show you some of this post-translation modification and epigenetic regulators to highlight the mechanism of HIF transcriptional activity in cancer cells. So recently, my lab identified the HIF-1 alpha lysine methylation. So this project was led by a talented postdoc, Lei Bao. So Lei used a purified HIF-1 alpha transactivation domain and incubated with a purified methyltransferases. So he found only Rna can methylate the HIF-1 alpha in vitro, but other transferases has no effect. So this methylation requires a Rna methyltransferase activity. So when he mutated this histidine to block the methyltransferase activity, he no longer sees the HIF-1 alpha lysine methylation. And he further identified the lysine 674 is the target of the Rna. So by mass spec, he further found the monomethylation and the dimethylation on these residues, but no trimethylation. So since this data has been published, so I just use a cartoon to summarize what we found. So the lysine 674 locates an inhibitor domain within the transactivation domain. So this then lysine 674 can monomethylate or dimethylate by Rna or the Rna paralog GLP. So this methylation can block the HIF transcriptional activity and glioblastoma cell migration. Interestingly, in glioblastoma, the Rna protein is lost. So the HIF-1 methylation is reduced, then can keep the highly active HIF-1 in glioblastoma to control the glioblastoma progression. So we also know that RNA-PO2 is essential for the gene transcription. So previous RNA-PO2 CHIP-seq studies show that RNA-PO2 pausing and releasing is essential for the HIF-dependent gene transcription. So the question is, what's the underlying mechanism? So we recently found chromatin remodeler called the CHD4 can interact with the HIF-1 alpha and the HIF-2 alpha. So here's the data from the breast cancer cell lines. From the using the core immunoprecipitation, we can find CHD4 can precipitate by either HIF-1 alpha or HIF-2 alpha or HIF-1 beta antibodies. Conversely, this HIF-1 alpha, HIF-2 alpha, and also can be precipitated by CHD4 antibody, suggesting this CHD4 can physically interact with HIF-1 and HIF-2 in breast cancer cells. And we further mapped their binding domain. We found CHD4 can predominantly bind to the transactivation domain of HIF1 alpha as well as a HIF2 alpha. So suggesting this CHD4 may play important role in HIF transcriptional activity. So the CHD4 is a key component of the noodle complex, which are known gene co-repressor complex. So then we, and the CHD4 also has a ATPase motif activity and also has a single finger PhD motif, which can control the gene transcription. So then we perform the HIF transcriptional activity using the luciferase-based transcriptional activity. So we found CHD4 can enhance the HIF1 transcriptional activity, but this is independent of the ATPase activity. So when we mutate this lysine residue on CHD4, we can still see the HIF activation. Conversely, when we knock down CHD4, we can reduce the HIF transcriptional activity. So we analyze what HIF target genes are controlled by CHD4. So here data from the Rna-Seq in the breast cancer cells. So you see a subset of CHD4 downstream target gene can overlap with the HIF target gene and hypoxia. And those target genes are involved in angiogenesis, hypoxia response, or the matrix reorganizations. So the question is how CHD4 control the HIF transcriptional activity. So by chromatin immunoprecipitation, we found CHD4 and HIF1 alpha can mutually recruit to the hypoxia response element. So you can see here in the those are HIF target genes. CHD4 was enriched near the transcription near the hypoxia response element, and this enrichment was enhanced by hypoxia. But this induction was blocked by knockout of HIF1 alpha and HIF2 alpha, suggesting this CHD4 was recruited by HIF1 and HIF2 to the HIF target genes. But this is not observed in the non-HIF target genes. On the other hand, we also found that HIF1 alpha binding to the HRE was blocked by a knockdown of the CHD4. So CHD4 can also recruit to the HIF1 to the downstream target genes. Then we analyzed the CHD4 and the Rna-Seq. We found the CHD4 can physically interact with the Rna-Seq, and this interaction was independent of oxygen tension. So then by chip chromatin immunoprecipitation, we saw knockdown of CHD4 can reduce the Rna-Seq binding to these HIF target genes, suggesting this CHD4 is required for Rna-Seq loading on the HIF target genes. So a previous study showed that histone acetyltransferase (CBP) is involved in the Rna-Seq recruitment to the target genes. So we wonder whether this also happened in the HIF target genes. So we saw that CHD4 can physically interact with the P300 and in the breast cancer cells. But when we knock down of P300, we found this Rna-Seq and the CHD4 binding is blocked, suggesting that P300 control the CHD4 mediated Rna-Seq recruitment to the HIF target genes. So what happened in the in tumor in vivo? So we found if we knock down CHD4, we can see reduce the tumor growth in orthotopic mouse models. Then in these tumors, we saw the a subset of HIF target genes reduced in the knockdown tumor. And we also see similar effects in MDA MB231 tumor models. And interestingly, when we re-express CHD4, we can rescue the tumor growth. Further, we found this CHD4 overexpression can increase the tumor growth, and this can be abolished by knockout of HIF-1 alpha and HIF-2 alpha, indicating that this HIF-1 is required for CHD4-mediated tumor growth. So to summarize what I've shown you of this part, so we found CHD4 can interact with P300, and the P300 can further recruit Rna-Seq. So these form the complex under normoxic conditions. Upon hypoxia, CHD4 can recruit the HIF-1 to the hypoxia response element. So this complex can to initiate the downstream target gene expression. So now the question is how the POR2 release to enhance the HIF transactivation. So we address this question by starting to analyze the hypoxia-induced epigenetic regulators in breast cancer cells. And we found the protein called Z1D8 from this microarray data. So Z1D8 gene is identified in early 2000, and it belongs to the bromodomain superfamily. So besides bromodomain, it also has a PhD domain and a PWP domain. So this typical the histone reader cassette can recognize several modified histones, so which are involved in the gene transcription. So the M1D domain, which locates at C terminals, is involved in the protein-protein interaction. So we characterize the Z1D8 in hypoxic breast cancer cells, and we found the Z1D8 is another HIF co-activator. So you see this Rna-Seq data. So you see many HIF downstream target genes are overlapped with the Z1D8 target genes. And those genes are involved in the glycolysis, angiogenesis, etc. And we further show this by chromatin IP sequencing. We see the HIF-1 alpha and the Z1D8, two proteins are co-localized across the human genomes at the hypoxia response element. So here just show you one example, show this HIF-1 alpha and Z1D8 can co-localize as a promoter of the HIF downstream target genes. So we did a lot of experiments to elucidate the mechanism of Z1D8 induced HIF transactivation. So we found under normoxic conditions, Z1D8 is associated with a hypoxia response element, and Z1D8 can acetylate by acetyltransferase P300. And this acetylation can recruit another bromodomain BRD4. So these three proteins form a complex under normoxic conditions. Upon hypoxia, HIF combines to the hypoxia response element and interact with this protein complex. So the BRD4 can activate the Rna-Seq to promote the elongation of HIF downstream target genes. So then we further characterize Z1D8 in the breast cancer cells, and we found Z1D8 is a very powerful oncogenic driver in breast tumors. So here the data from the orthotopic mouse model. So when we knock down Z1D8, we can see the reduced tumor growth. And this can be partially rescued by wild-type Z1D8, but not this lysine mutant, which is an acetylation-resistant Z1D8, suggesting this HIF-1 is required for Z1D8-induced breast tumor growth. And in these tumors, we also see the increase in the knockdown tumors. We see the increased cleaved caspase-3 and reduced angiogenesis. So this can be reversed by wild-type, but not an acetylation-resistant Z1D8. So here's the data quantification, and a similar phenotype for the lung metastasis. So suggesting this Z1D8 can control the breast tumor growth and metastasis through activation of HIF-1 and HIF-2. Then what about human patients? So we analyzed several databases, including this TCGA data. So you see that about 40% of human breast cancer patients highly express the MRD8 gene. And we also validate this mRNA expression in the triple-negative breast cancer tumor microarray. So you see the MRD8 protein is strongly expressed in the triple-negative breast cancer. And in contrast, this adjacent normal breast tissue don't express or weakly express MRD8 protein. And we also show this MRD8 is connectively correlated with a post-survival of breast cancer patients. So now in the next few minutes, I'm going to talk about the HIF downstream target genes. And we know that HIF control many downstream protein-coded RNAs, which are involved in the those cancer biology, as I mentioned. We also know that about third quarter of the RNA are transcribed to the and the majority transcribed RNA belongs to the non-coding RNAs, and non-coding RNA is one of the this non-coding RNAs. So which is a class of transcribed RNA with more than 200 nucleotides. So we are pretty interested in this non-coding RNA because its function is really unknown in human cancers. So from the gene expression data, you can see that in human cancers, the non-coding RNA is highly dysregulated. So the number of the dysregulated non-coding RNA is comparable to those of the protein coding RNAs. And the recent studies show this non-coding RNA play very important role in cancer biology. So we ask an important question whether hypoxia can control the non-coding RNA in breast cancers. So my former postdoc, Yan Lin, did a transcriptional analysis. So using this two breast cancer cell lines, MB231 and SUM159. So she identified over 100 non-coding RNA, which are induced by hypoxia. So from these two cancer cell lines, she found 45 non-coding RNA are overlapped. So we focus on those uncharacterized non-coding RNAs. And here, today I just show you one of example of this RAB11B and sense1. So RAB11B and sense1 is an antisense of the RAB11B gene and located on the chromatin 19. So we confirm the RAB11B and sense1 expression in the breast cancer cells using this race experiment. And we further show this non-coding RNA can be induced by hypoxia. And importantly, when we knock out a HIF2 alpha, we no longer see the hypoxia induction. In contrast, HIF1 alpha knockout has no effect, suggesting this non-coding RNA is a HIF2 target genes. So this non-coding RNA is also induced in human breast tumors. And importantly, this non-coding RNA can significantly correlate with the HIF target genes like VEGF-A and angiopoietin-like 4. So then the question is, what's the function of this non-coding RNA in human breast tumors? So we generated the RAB11B and sense1 overexpressed breast cancer cells. Then we tested their tumor growth in the orthotopic mouse model. So we found this RAB11B and sense1 has no effect on the tumor growth. But we see the increased lung metastasis and as well as a liver metastasis. Conversely, when we knock down of this non-coding RNA, we see reduced metastasis to the lungs and the liver. So suggesting that this non-coding RNA is specifically control the distance metastasis in mice. And in these tumors, we found angiogenesis is increased in this RAB11B and sense1 overexpressed tumors. In contrast, this Ki67 was not effect, suggesting this link RNA has no effect on the breast tumor proliferation. So we know the tumor angiogenesis is linked to the breast tumor metastasis. So this angiogenesis is regulated by angiogenetic factors, which are also controlled by hypoxia. So we wonder whether this LAB11B and SENS1 can control the angiogenic factors to increase the tumor angiogenesis and metastasis. So we did an Rna-Seq experiment to identify which angiogenic factors are regulated by this non-coding. So we found in this experiment we found over 1,500 genes was induced by this non-coding RNA. On the other hand, this over 1,600 genes was downregulated. So within these upregulated genes, we found a subset of angiogenetic factors and also hypoxia-inducible genes. So then we did the real-time PCR to validate those angiogenic factors. So you see the VEGF-A and the angiogen protein like 4 was induced when we overexpress this LAB11B and SENS1. Conversely, when we knock down LAB11B and SENS1, we can see this angiogenic factors was inhibited under hypoxia conditions. So suggesting that this LAB11B and SENS1 can control this VEGF-A and angiogen protein like 4 to increase the angiogenesis. So now question is how the LAB11B and SENS1 regulates angiogenic factor expression. So we know the non-coding RNA can interact with the transcription factor to control the gene transcription. So as I mentioned, those angiogenic factors are regulated by HIF, but we found the LAB11B and SENS1 does not interact with the HIF. So then we look at the RNA-PO2. So since the non-coding RNA also control the RNA-PO2 activity. So by an RNA pull-down assay, we found the LAB11B and SENS1 can pull down the RNA-PO2, and again, this interaction was independent of oxygen levels. When we knock down LAB11B and SENS1, RNA-PO2 binding to these angiogenic factors was reduced, suggesting this LAB11B and SENS1 can recruit RNA-PO2 to control this angiogenic factor expression in breast cancer cells. So here in this project, we've identified the LAB11B and SENS1 is a hypoxia-inducible gene, non-coding RNAs, and this is exclusively induced by HIF2. And the LAB11B can interact with the RNA-PO2 to induce the angiogenic factor expression. And those angiogenic factors can control the tumor angiogenesis and the distance metastasis. So we show this LAB11B and SENS1 is a hypoxia-inducible trans-acting non-coding RNA in breast cancer cells. So finally, I would like to thank our lab members who did this work. So I already cited their name. So I also want to thank our collaborators at the UT Southwestern, and also many thanks for our funding agencies. And also thank you for your attention. I'm happy to take your questions.
Thank you so much, Dr. Weibo. Yes, any questions from audience? If not, we will go ahead with the next presentation.
Yeah, sorry. Hi, could I ask a question?
Yes.
I was wondering, do any of these factors that interact with HIF1 alpha modulate the stability of HIF1 alpha per se?
So those epigenetic regulators I just presented has no effect on the HIF1 alpha protein stability. They just control the transcriptional activity.
Okay, so their interaction does not result in stabilization of the protein?
No. So
okay,
like a PhD, yes, but not the CHD4 and the Z1D8.
Okay, so when you overexpress these factors in the tumor in normoxia, it doesn't change the level of HIF1 alpha?
No,
okay.
No, not at all. So it's only, yeah, as I said, it could just control the transcriptional activity.
Okay.
Nothing to do with the protein stability.
Thank you.
Thanks.
Yes, thank you, Dr. Weibo.